Learners, welcome to today's Life Sciences lesson. My name is Notlanhla Mtshali. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Today is the continuation of last week's lessons, which is genetic and inheritance, which is what you are currently busy with now in your schools. So what we are going to look into today are the 4 topics, which is the dihybrid crosses, pedigree diagrams, mutations, and genetic engineering. Okay, first things first, we are always going to refer you to your exam guidelines, so just bear with us. So we need to go and check what does the exam guideline tell us about the dihybrid crosses. So under dihybrid crosses, reminder that we need to look into the Mendel's principle of independent assortment, which states that the various factors controlling different characteristics are separate entities and do not influence each other in any way and sorting themselves out independently during gamete formation. We're also going to look into what dihybrid genetic problems. So we're going to solve dihybrid genetic problems. We're also going to look at the determination of what? Determination of genotypes and phenotypes, proportion, percentages, and ratios. So last week what we did were monohybrid crosses, but now we're looking into what is called dihybrid crosses. So what is a dihybrid cross? Dihybrid cross, the word di, from remember diploid, all those dyes that we did earlier in the year, diploid meaning 2. So this is a type of the cross that involves 2 traits, or it involves 2 characteristics. So the other one that you did last week, it was a monohybrid involving 1 characteristic. So here we are looking at 2 characteristics at a time. Why? So that we go and look at the principle, because remember this Mendel's law of independent assortment tells us that they separate individually, they do not influence each other in any way, and they can be different, they can be present in different gametes in any way. So why are we looking at the 2? So that when meiosis takes place, we look into how they separate and how they form gametes and how they are in different gametes. So the law of independent assortment says that alleles of a gene of 1 characteristic. So alleles of a gene of 1 characteristic, they segregate independently of the alleles of the gene of another characteristic. And then remember that we say that the genes are found where? On the chromosomes. And then during meiosis, when the chromosome separates, that's when we talk about these alleles separating independently. So another thing that you need to remember also with the dihybrid crosses, which is very important, is that when you cross a heterozygous organism with another heterozygous organism for both traits, you will always get the following phenotypic ratio. This always stands. So in other words, we said we are looking at 2 characteristics at a time. So the diagram that we have on the right here, we're looking at the color of the seeds and also the shape, the seed and the shape. So it's 2 things at the same time. So when you cross these characteristics and they are heterozygous, at all times you're going to get the ratio that is 9:3:3:1. So let's put this into practice. Let's look into a question, for example. So we're going to look into this one, which says in plants, tall and yellow flowers are dominant over short and orange flowers. A plant with heterozygous for both traits is crossed with a plant that is heterozygous for tall and with what? With orange. Okay, I'm gonna go back, sorry. Okay, start again. So we're looking at a plant, or in plants, we're looking at 2 characteristics, the tallness or the height of the plant, and we're also looking at the what? At the color of the plant. So those are the 2 characteristics that we are focusing on. And then we are told that tall is dominant over short and yellow is dominant over what? Over orange. So now the statement says a plant which is heterozygous for both traits. So we're looking at the plant that is heterozygous for tall and heterozygous for color, which is yellow. Remember, when it's heterozygous, it is 2 contrasting characteristics. and then it is crossed with a plant that is heterozygous tall, so it will have big T, small t, and with what? With orange flowers. We were told that orange flowers are recessive, so they can only be orange if it's two small letter Y's. Remember, as soon as you see a capital letter and you are told that it's a dominant, so here this orange color is brought by two recessive alleles. In this case, there will be two small Y's. So this is the cross that we are going to be looking into. So we are going to do a genetic cross to show the possible offsprings of these parents. So parent number 1 is heterozygous for both traits. So tall and with yellow flowers crossed with tall and orange flowers. So let's look at the genetic cross. So what do we do? Step number 1, you identify the phenotypes of the two organisms for which the two characteristics that will be crossed. So we've already identified that. We said we are looking at the tall yellow flowers with the plant with tall and orange flowers. Then step number 2, then we are going to look into the letters. In this case, we were given the letters in the statement. Sometimes letters are not given, you will have to come up with your own, but most of the time you will be given the letters. So we were told that capital letter T is for tall and small letter t represents short, and then capital letter Y represents yellow and small letter y represents orange. So those are the letters that we are going to use to represent the genetic makeup of the two parents. And then step number 3, then we write down the genotypes of the parents. So as we've indicated earlier, the first one, we were told that it is heterozygous tall and heterozygous yellow because it's heterozygous for both characteristics. The second one is heterozygous tall, but the flowers are orange. So it is homozygous orange for the flowers. So then now we have step number 3, where we've identified the genetic makeup of the parents. Then moving on to step number 4. Now we're going to try and determine the gametes. So how are these alleles? Remember here, this is in a cell, this is also in a cell, and then meiosis is going to take place here, and then these must now separate independently into different gametes, which is what we called earlier Mendel's principle of independent assortment, where an allele of one gene could appear in the same gamete with any of the alleles for the other gene. So then we determine the possible gametes. So now, grade 12 learners, do not be confused. When we work with the dihybrid crosses, in order to determine the gametes, the possible gametes of this parent and the possible gametes of this parent, we always go back and use the Punnett square because it's the easier method. But I know that some of you also prefer to use what is called the foil method, where you will cross the first one and the inside one and so on until you get all the gametes. So you can do that one if you want to, or you can just simply draw a Punnett square. So if you look at the first set of parents, the gametes that are possible from this parent are capital letter T, capital letter Y, capital letter T, small letter T, big Y, and small t and small y. Because remember, if we separate this, so if this crossed with that, so these alleles can pair up. There's a possibility that the small y can pair up with the small t. There's also a possibility that big T can pair up with small y, or big T pairs up with another small y. So this is the independent assortment that we are referring to. So these are the possible gametes from parent number 1. And then again, we work out the possible gametes for parent number 2. Same thing happens. Different possibilities here. So this is one possible gamete, big T, small y. Another possible gamete, small t, small y, big T, small y, and small t, small y. So then these are all the gametes that are possible that can come up of these two parents. Then the next thing now is to then determine, remember the question says determine the possible genotype and the phenotypic ratio. So how are we going to do that? The same way that we did when we're doing the monohybrid crosses, we're going to draw a Punnett square. But this Punnett square is going to be different from the other one because with your monohybrids cross, the Punnett square is very small. But this one is a Punnett square that is sort of a 4x4. So you need like 16 columns at the end, so that you must put all your gametes there. So the gametes from the first parent are gonna go all there at the top, and then the other parent gametes are gonna go on this side. And then the same thing that we did with the monohybrid. So you crossed this one and that one, and then you start filling in the first row. So with your first one, the second row, the third row, the fourth row, up until you filled up all the columns in here to figure out what are the possible genotypes. So let's look at how it's going to look like. So remember, random fertilization of gametes from both parents could fuse in different combinations to form the offsprings. So then you write down your gametes. So the answer would look like this. So these were the initial gametes that we wrote, and then the possible offsprings are now, if this and that is crossed, we have big T, big Y, small y. And then if we continue with this one, it's big T, small y, small y. We have big T, small y, and then the last one is big T, small y, and big T, small t, and big Y and a small y. So then you're going to continue with this pattern until you filled up all the possible genotypes. So then what will be the next step? The next step is now to look into the phenotype. Okay, before we go into the phenotype, if you look just at the genotype, we can already identify how many plants are tall, with how many with yellow flowers. So we have this one because of the big T and the big Y. So obviously here it's tall with yellow flowers. Also this one is tall with yellow flowers, tall with yellow flowers, tall with yellow flowers, tall with yellow flowers, another tall with yellow flowers. So the phenotype, we have 6 that are tall with yellow flowers. And then if you look at another one, we're looking at tall with orange flowers, tall with orange flowers, tall with orange flowers, tall with orange flowers, another tall with orange flowers, and another tall with orange flowers. So again, if you count, we have 6 tall plants with orange flowers. And then let us see, what do we have left? We have those ones that are short and orange flower. So we have 1 short orange flower, 2 short orange flower. So then we have 2 that are short with orange flowers. And then lastly, we have 1 with short and yellow flowers, and then another one with short and yellow flowers. So we have 2 that are short with yellow flowers. So the phenotypic ratio here will be 6:6:2:2, because we have 6 that are tall and yellow with 6 that are tall and orange, is to 2 that are short and orange, and is to 2 that are short and yellow. So let's see what the answer looks like. So this is now what the question wanted us to do, to determine the phenotypes of the offsprings from the genotypes obtained in the Punnett square. So we have 6 tall and yellow, 6 tall and orange, 2 short and yellow, and 2 short and orange. We are going to take a 2-minute break. We'll come back soon.
Dreams. What is your dream? Yini iphupho lakho? Not your parents' dreams, not your mom's dream for your life. What is your dream? Is it to get a bursary? Is it to study science? Is it to become a DJ? Is it to become a singer? What is your dream? Iphupho lakho. Once you have that dream, what I need you to do, I need you to write it down. Ingole mofati, write it.
The road to success, to becoming a successful student, is through commitment. Commitment to studying, commitment to putting in the extra hours in your books. I know it's not going to be easy. Actually, no one said it will be easy. But if you keep pushing and if you keep believing a little more in yourself, then the results will show in your life. Commit and become. The road to success is to commit to yourself. In order for you to become who you want to become as a student, you need to commit to reading your books, to studying, to bettering yourself. I know it's not going to be easy. Actually, no one said it's going to be easy. But if you put in the work, the results will show in your life. Tap into your power. Sometimes you will feel like putting away your books, and not reading as much as you're supposed to be. But I want you to commit, because through commitment, there is infinite power.
Dreams. What is your dream? Yini iphupho lakho? Not your parents' dreams, not your mom's dream for your life. What is your dream? Is it to get a bursary? Is it to study science? Is it to become a DJ? Is it to become a singer? What is your dream? Iphupho lakho. Once you have that dream, what I need you to do, I need you to write it down. Ingole mofati. Write it down, ibhale phansi, and read it each and every single day when you wake up. Read it each and every single day before you go to bed, and remind yourself of where you want to see yourself. What is your dream? Write it down, meditate on it, and go after it with everything that you have.
Please just keep an eye there, Nene, because she does faint. Right, welcome back. Now that we've done that example on the yellow and orange flower plants, let's do another one on animals. It's important to remember that when it comes to dihybrid crosses or monohybrid crosses, we can look at any organism. So we've done one on plants, so let's have a look at one on animals. These are about rabbits. In rabbits, we have gray fur, capital G is dominant to small letter G, white fur. Also in rabbits, black eyes, capital B, are dominant to red eyes, small b. A rabbit with gray fur and black eyes is crossed with a rabbit with white fur and red eyes. Both parents are homozygous. The F1 offspring were interbred. Use a diagrammatic cross to show the genotype and phenotype of the F2 generation as well as the phenotypic ratio. So when you're looking at this type of a cross, even with your monohybrid cross, it's very important that you underline or highlight the instruction. What is the question instructing you to do? So we've got a beginning part, which is the information or the stem of the question that's sharing information with you. And then we've got the second part, which is giving you the instruction of what needs to be done. So they're saying that we had both parents were homozygous, okay? And the one was gray with black eyes and the other one was with red eyes. So let's think now, what was that P1 generation, okay? What were the genotypes for that? And you would see that we would come to, for the one parent, and then the other parent, okay, homozygous. So it's homozygous dominant for the one parent, homozygous recessive for the other parent. And they're saying that these ones had offspring. So the F1 of the F1 offspring, what would they be? So little trick, you can do the whole cross if you want to, but there's a little trick to figure it out because of the law of dominance. So the law of dominance says that if we have a homozygous dominant individual that's crossed with a homozygous recessive individual, all the offspring in that generation will be heterozygous for both traits. So now they're saying in the question that this F1, these were interbred. So now we've got to show what is the F2 generation. So this one becomes the P1. So the P1 becomes, right? And then now we need to, the P2, sorry, this is the P2 now. This was the P1. The first generation, then we had the F1, then they were interbred, so we have the F2, and now we need to work out what is the P2. We have to work out what is the F2. Okay, so we've got, let's work through the answer.
So like we first said, we first have to identify the P2 phenotype. It was gray fur. We didn't speak about that. Okay, let's go back quickly. What are the phenotypes of these individuals now? It will have black fur and have black eyes. Okay? Right. So here we have gray fur and black eyes. So let's just go back there. Sorry, it's gray fur. It's the capital G, the dominant elite is the gray fur. So it will be the gray fur with the black eyes. So that's the one, both organisms are the same. Both parents will be gray with black eyes. And the genotype, we spoke about it, the heterozygous for both traits, capital G, small g, capital B, small b, and those are the parents. Then we have meiosis, okay? And then after meiosis, we've got the gametes, okay? How did we work out the gametes? As Nomtanta showed you earlier, you can do a little Punnett square for each of them as long as you know that each gamete is going to have one letter of each trait. So that's like a tick box for you when you're doing this to think, how do I know that I'm on the right track? When you are forming the genotype, you must make sure that you have 2 letters for each trait. When you are doing the gametes, you're going to have 1 letter for each trait. Okay, so as you can see it over here, it is all like that. Then we've got fertilization. Okay, and then when fertilization occurs, that's when those gametes are going to fuse. And now we've got the F2 genotype. And now you're going to work out the Punnett square. Okay, so here we have the big Punnett square. We're going to put the gametes in here. We can actually write it there. The gametes there. And the gametes, so we're going to put them this way and this way. And then you're going to do your normal Punnett square. And then you're going to get all of these. So as you can see over here, they are color-coded. Okay, they are color-coded because this is easy for you to do because remember they want to know the phenotypic ratio. If we go back to the question, they asked us for the phenotypic ratio. So we have to look at all the different, count the same phenotypes. So even though the genotype is different, remember the phenotype could be the same because of the law of dominance. So let's have a look at the first one. This one is gray. It's gray with black eyes. This one is also gray with black eyes. All the ones that are in black are gray with black eyes. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so we already know that we have 9 of those. And then we've got the next color. Let's see, this one is gray with red eyes. 1, 2, 3. Then the next one is going to be white with black eyes. 1, 2, 3. And then the last one is going to be white with red eyes, which is 1. So we have to work out what is the ratio. Ratio, we cannot write it like that. We have to write, first we need to write out the phenotype, which we just did. Then we need to put the ratio, gray, black, red, white, black, white, red. And then we come up with the ratio 9, 3, 3, 1. Okay, and as we mentioned earlier, that is our ratio that we would have if we have to cross 2 heterozygous individuals for both traits. We would have all the offspring having the phenotypic ratio 9:3:3:1. Right, so now you should be getting a little bit better at this. Okay, now we're going to go through a little question. Okay, that is going to discuss another one. So they won't really give you a dihybrid cross to do where you have to work out all the steps for 1 mark. However, they can give you a question like this question here where they give you a scenario and they ask you different questions on them. They could ask you for the phenotypes of the parents. They could ask you for the offspring. They could ask you these questions that are coming here. So you don't have to do the whole cross, but you're going to have to do parts of the cross in order to answer the questions. So let's have a look at this one. In summer squash plants, white fruit color, capital B, is dominant over yellow fruit color, small b. And round fruit, big D, is dominant over oval fruit, small d. A summer squash plant that is homozygous for white and round fruit is crossed with a plant that is homozygous for yellow and oval fruit. State the genotypes of the P1 parents. Okay, so now they're saying homozygous white and round, okay, with homozygous yellow. So we are going to have the situation where we are going to have the homozygous for both traits. Okay, so we have capital B, capital B, capital D, capital D, small b, small d, small d. Okay. Then the phenotypes of the F1 generation. So now we're saying what are their individuals, their offspring going to be. So it's similar to the previous one we did when I said that if we have homozygous dominant with the homozygous recessive, all the offspring are going to be heterozygous, okay? But they're not asking for the genotype, they're asking for the phenotype. So how do you work this out? You would work out what the genotype is, okay, which we spoke about heterozygous for both. Therefore, it has to be white and round fruit. Please take note that with this one, it's 2 marks all or nothing, which means that you're not getting 1 mark for saying white and 1 mark for saying round fruit. The whole phenotype is given 2 marks. So when it comes to genetic problems, don't really look at the mark allocation to indicate to you how many words you should have or how many facts you should have. Okay, just answer the question correctly. 2 plants that are heterozygous for both characteristics were crossed. So like I said, it's like the previous one we did. So now we've got ones that were both heterozygous, and we have to give all the possible genotypes in the gametes that will be formed. Okay? So now we have to work out the gametes. So you can do your little crosses, and then we'll get your answer. Big D, big B, small d, big B, big D, small b, big D, etc. Okay, so it's similar to the previous one. Just make sure that you have each letter from each trait, so you have 2 different letters. And then you apply the law of independent assortment. How many plants in the next generation are likely to have yellow and oval fruit? Okay, so now they're saying that now how in this generation, how many would have yellow and oval fruit? So remember, those are the homozygous conditions. And what is the ratio again when we cross the heterozygous with the heterozygous? 9:3:3:1. And if you can remember correctly, the homozygous recessive individuals for both traits is 1. Therefore, the answer is 1. Give the possible genotypes of both parents that must be crossed if a farmer wants summer squash that are white with oval fruit only. So now he doesn't want he wants white fruit. So the capital B has to be somewhere. And he wants oval fruit. Let's see. Right, he wants oval fruit. So the oval fruit will be the small letter D. So then we know that in the offspring, okay, they definitely, for in order to have oval fruit, you have to have homozygous recessive. Okay, and from what you know, we need to, if it's going to be homozygous recessive, remember they must get 1 trait from each parent. Okay, 1 allele, from each parent, which means that the parents must be having a small d. Okay, definitely. So it can either be, these are different parents. We can either have homozygous dominant with homozygous recessive. Okay, so these are the 2 parents. Both of the parents can be like this. You'll see if you do the cross that you're going to be able to get, all of them are going to have white fruit color because we only have a capital B there. And then we've got the small d so that we can ensure that we have a homozygous recessive, which will give us the oval fruit. And then this one is also possible. Why? Why is this one possible? Because remember that a capital B with a small b is still going to have the dominant characteristic that's going to be in the offspring, which is in this case white fruit color. So no matter how you're going to cross this, you're still going to get individuals that are going to be either this or this. This is the offspring that we want, okay? Because these offspring are all white and oval. Okay, white fruit color, and it's oval, right? Then the last one is also possible because even with this one's a little bit more tricky to remember, but even with this one, you would still get it because here we have 2 capital B's, you see. So because of the law of independent assortment, once you do all of your gametes and all of that, you will figure out that you do actually get to that answer. Okay, so we've done enough dihybrid crosses now. Let's go on to our pedigree diagram, our next part, which is going to be our pedigree diagrams. Okay, let's get right into it. The main thing I just want to summarize about dihybrid crosses is that we really need to have a look at, keep your rules, know your rules. Let's just go over it quickly. The main thing about dihybrid crosses is that there are 2 traits. Okay, so we have 2 traits, whereas in monohybrid we only have 1 trait. All the other things that you do for the monohybrid cross is still going to apply. You are still going to use the same recipe. Okay? The thing that differs here is that in your phenotype, you are going to mention 2 traits. In your genotype, you are going to have 2 letters, 2 different letters. You're going to have 4 letters in total, 2 for each trait. Okay? But you're going to have 2 different letters. Then the next thing you need to remember is that when you are looking at your gametes, that each gamete must have one of the alleles of each trait. So you're going to have 2 different letters. Okay, so once you remember those things, check yourself again and tell yourself, okay, do I have this? Do I have that? Remember those little tips or those little rules so that when you're getting dihybrid questions, you'll be able to answer any question that is given to you. Also remember your law of independent assortment and your law of dominance. Okay, those things also help you to answer some application questions. Now we go on to genetic lineages, the pedigree diagrams. Okay, and basically these trace inheritance of characteristics over many generations. It's basically your family tree. Okay, so we've got a family tree over here. As you can see, we have a family tree. You can see there's grandma and grandpa, and on both sides of the family. Then we have mom and dad, and we have all the children. This is the family tree. Okay, this is that up, and it's also our pedigree diagram. The pedigree diagram is going to show the inheritance of different characteristics or genetic disorders that are present in a particular family. So when you're looking at genetic crosses, in pedigree diagrams, you have to have a look at the shapes. This whole diagram, the shapes, the vertical line, the horizontal line, the squares, all of those things mean something, and you need to know what they stand for. So the circles are female, the squares are males, and the horizontal line between the two is the marriage line. The vertical line going down is the line of descent, and that leads to their children. And then we've got this little box line over here, which shows the siblings. Okay? And then we also have shaded and unshaded. If it is unshaded, it is the person that is unaffected with the trait, and the shaded is the affected with the trait. But never just remember this by face value. Always look at the key. The key will tell you or guide you when you're given a cross like this. The key will guide you on what it is that you need to say. Then we mustn't forget about the generations. You must be able to count the generations, okay? So the main thing about this is count the rows. That's the way I always remember it, is that I count the rows. So we look the first circles and circles and squares that you see is going to be the first generation. Those parents' children are the second generation. Then when those children have children, that is the next generation. That's why we count every row. So the first generation are the first group of parents to have offspring in the particular scenario that you've been given. Okay. Right, so let's just have a look at this again. Here we've got 3 generations. Like I said, here we're looking at this one, there's 3 generations. How many males can we see? Okay, we can see 1, 2, 3, 4 males, and one of them is affected. Okay, we've got how many females? 1, 2, 3, 4, 5 females, and 3 of them are affected. How do we know that? Because the affected ones are the ones that are shaded, as I mentioned before. Right. Now we have to, this is the steps in solving a pedigree diagram. Now you've been given this diagram, you're given a key, and as you can see, there are no genotypes here. But what is the key giving us? There's something I forgot to mention. The key is giving us the phenotypes. So these are all the phenotypes. Okay, the key has given us the phenotypes. The key is also telling us that the shaded ones are the males with the attached earlobes. And the unshaded ones are the people with the unshaded is with the unattached earlobes. Okay. But they're not telling us which one is the dominant allele or which one is the recessive allele. Okay. So, but they've got a note there for us. They will always give you an indication. So in this instance, they didn't give us some genotypes in here that would have given us an indication. But here they told us that unattached earlobes are dominant and attached earlobes are recessive. We must complete the missing genotypes. Okay, so the ones we can definitely see then are the shaded ones. The shaded ones are the ones that we can already tell. The shaded ones are the ones with the attached earlobes. So we already know that if they're going to have attached earlobes, they must be homozygous recessive. So we can already fill in the genotype of all the homozygous recessive ones. So whenever you get a pedigree diagram, work out the offspring, okay? So there we've got, we're giving you the steps here. If you study the key, you look for the dominant and recessive characteristics. We did that. We fill in the genotype of individuals with the recessive condition. Okay, so here we're going to fill them in. Then each individual with the recessive condition inherits one allele from each parent. I just want all the steps to come up and then we can go through it. Sorry. Right. So the first thing so that's what you're going to do first. You're going to put in all the small f's. Okay, so let's just quickly put them in here. Those are the ones that you know. So then you're going to work backwards and fill in the one recessive allele for each parent, because you know that if the offspring must have the recessive condition and it's homozygous recessive, each of those recessive alleles must have come from the parent. So one will come from each parent. So then you're going to work it back. So this parent, one F will come from this parent and one F will come from that parent. So that parent definitely has to have a small f. But this parent has unattached earlobes, which means that they also need to have a capital F. You see? So this one is heterozygous. So that's how you basically work through it, and then you work out all the other ones that you can identify based on using this principle. You first allocate all the homozygous recessive ones, and then you know that if the offspring is homozygous recessive, must have received one allele from each parent, one of those recessive alleles from each parent, and then you work back. Okay. Now we did that previous example, so we're going to move on to this one. Okay. The pedigree now. The previous one we did, I just want to show you this one as well. The first one we did was unattached and attached earlobes. Okay, so you see that it's a normal inheritance. And we're not doing dihybrid. Can you see that these are monohybrid crosses now? Okay, so we are looking at one trait. So the previous one was attached and unattached earlobe. This is the example of another one. Okay, this has got to do with the lips. And here we've got broad lips and thin lips. Okay, so it's another characteristic, and it is also it's complete dominance, and it's also a monohybrid cross. Okay, but now so you have to look very carefully what are they giving you, because now this one, for example, is on color blindness. And what do you know about color blindness? Is that it is sex linked. Yes, it is sex linked inheritance. Okay. So here we have a pedigree diagram for color blindness. We must determine the possible genotypes for numbers 1 to 11. The color blindness is a sex-linked condition. We must use this annotation. It's X to the small letter b to indicate the affected allele and X to the big letter B to indicate the normal allele. Okay? So let's look at our key. So our phenotypes are telling us that the shaded ones are the affected males and the unshaded ones are the normal males. So let's first ask ourselves, okay, let's first put in the X's and the Y's, because you know when you did sex-linked inheritance with your normal monohybrid crosses, that was the first thing that you did. Okay, so let's put in all of the... So that's a female, so to be XX, male, XY. This one is the female, XX, male, XY. This is also a male, XY. This is a female. This is a male, XY. This is a female. Right. We're going to fill in all of them. So now they're saying that it is, okay, this one is, they're using this one to the affected allele, which means that it is carried on the recessive allele. Okay, it's very important that you indicate that, because some disorders are carried on the dominant allele, but this one is on the recessive allele. Okay, so now at least we know that. Now we go to the affected male. All the affected males, remember they have an X and a Y. So in order for the male to be affected, which letter are they going to need? Which allele are they going to get? They're going to get this allele. Yes, the X to the small letter B. So we can already fill that in over here with all of the males that are affected. So we go to all the shaded males and we put in a small letter B for them. Okay, so we know that they are definitely going to be having the characteristic, and that's definitely going to be their genotype. We don't put anything on the Ys. Okay, you know that. Now you can work out all the females that are affected, because all the affected females. Oh, in this instance, we have no affected females. This one's making it a bit difficult for us here. Okay, but we're going to work through it. So now we have no females here that are affected, but we can still work it back. Now we can do the males. Let's look at the normal males. So the normal males will have this allele. Okay, so this one will be a capital B. Which another number? We have number 11, which will also have a capital B. Right. So now we need to account for number 1, number 3, number 6, number 8, and number 10. And all of these individuals are female and they are normal. So they do not have color blindness. But that doesn't mean, from what you know from the sex-linked inheritance, it does not mean that they cannot have the recessive allele. Okay. So how do we know? We look at the offspring. Let's look at this male. Let's look at number 5 first and number 7. Then they're easy to work back. Okay, so this number 5, males in general, number 5 is a male. Males can only receive an X chromosome from the mother. Why? Because the Y chromosome is coming from the father. So this B, this X to the B, it had to come from somewhere. And it cannot come from the father because the father gave this Y. Therefore, one of these alleles for the mother must be a small letter B. And I'm putting it on the second one. Why? Because the mother is unaffected, which means that the mother does not have color blindness, which means that the other allele must be a capital B. You see that, right? So then it works out, okay? Here that we have this one, and this one is also unaffected. But now let's look at the female. Because the female must get an allele from the mother and the father, the female, the daughter can obviously not get a Y from the father, because then she would be a son and not a daughter. So she could only have gotten this allele from the father. You see? So then we're going to have definitely one capital B over here. And the other one also has to be a capital B, because this one, it got one capital B, yes. But it can either get another capital B from the mother, okay? And then, yes, it got another capital B from the mother because the son got that one, right? But it could also get a small letter B. It would still be unaffected. Okay, so that's how you work them out. You're going to work all of them out like that, and let's have a look at our answers to see if we were correct. And you will see there that we've worked it out. Yes. Okay, so that's how we basically work out the genetic cross. You just do all of them, and then you work out what the offspring would be. Right, for all the offspring, and then you work out what the parents are. Okay, let's do one more pedigree diagram. This is one on Galt's syndrome. It's a sex-linked genetic disorder. It is caused by a dominant allele, X to the power of capital G. The diagram below shows the inheritance of Galt's syndrome in a family. Right, so here we've got the affected and the unaffected. Okay, once again, they're telling us affected and unaffected. I just want to talk about this quickly. Always use the phenotypes that are given to you in this block. Okay, always use the key that was given to you there. If they talk about affected and unaffected, then you talk about it. If they talk about specific things, if they say colorblind male, colorblind female, then you have to mention the exact word that they mention. Okay. So name this type of diagram. They love to ask this question. This is the pedigree diagram. How many females are in this family? So now we're just counting how many females there are. So you count all the circles. Here there are 4. No, sorry, there are 6. We cannot just count the circles. Here we have 1, 2, 3, 4 circles, but then we've got 2 females down here. They would also be circles, so there are 6. And how many males in the F1 generation have Galt's syndrome? Okay, they told us it is sex-linked, which means we're going to have X's and Y's. And how many of them have Galt's syndrome? It is 1, because there's only one shaded block, square. Now we need to give Gabby's genotype. So now here's Gabby. Okay, Gabby is a female. and Gabby is affected. Okay, so Gabby has the characteristics. Let's just go back to say, you see, it's caused by a dominant allele. It's caused by the dominant allele. So Gabby only needs to have one allele to be affected by the characteristic. Okay, Gabby only needs to have one allele. So this one is definitely a capital G. Okay, so we work it out, and it's going to be capital G and a small letter G. Why? Because the offspring, can you see that there are females that are unaffected, and the unaffected females will have to have homozygous recessive, and the homozygous recessive gene has to come from each parent. Okay, so one has to come from here and one has to come from this side. Okay, because this father Pit is going to be unaffected. Okay, so that this one is this person is also going to be unaffected, so it has to be looking like that. So this is where learners can get confused, because now you have to think to yourself, okay, this is caused by a dominant allele, not a recessive allele that we mostly get, or we mostly think or assume. Okay, so that's how we work out what Gabby's genotype is. Now, Anju and Pilusa, they have 4 children. Now we must give the phenotype of their sons. Okay, so they had 4 children. So we can see here there's males, right? We have to give the phenotype of them. They are going to be unaffected with Galt syndrome. Okay, why? Because remember, the father is going to give the Y, and the father is Pilusa. Okay, the father is affected, but the mother is unaffected. Okay, and then so the Anju will be able to give the unaffected allele to the 2 boys. Explain your answer. Now we need to explain, not the way I was explaining it. Okay, let's explain it in proper life science terms. We have to first, what you do is you state what the mother is and you state what the father is. Firstly, we're starting with the mother. Pilusa is affected. Anju is unaffected. Okay, so if they're unaffected, they need to have the small letter G. Remember, we said that, right? And Pilusa is affected, he has the capital letter G. Males inherit the Y chromosome from the father. So the boys will inherit the Y chromosome from Pilusa, their father. And they will inherit the recessive allele from Anju, because they have to receive, there's only a recessive allele there for them to get. So they will get the recessive allele. Okay, when you're answering explain question, just answer it in an explain and cause and effect manner. You state what it is there and how you get to your answer. Just write your steps, okay, one by one. The diagram below shows the pattern of inheritance of a disorder. Okay, so we will say male not affected, female not affected, and there's a female that is affected. One can conclude that this disorder is caused by, okay, so what are we looking at here? Is it caused by a recessive allele with both parents are heterozygous? Is it caused by a dominant allele with both parents homozygous? Dominant allele with both parents heterozygous. Recessive allele with one parent homozygous recessive while the other is heterozygous. Or the dominant allele with one parent heterozygous while the other is homozygous recessive. So they're showing us here that the female is affected. Okay, so it has to be the first one where they say the recessive allele with both heterozygous parents, because let's try to work it out. Let's just make a letter here. If these two are this is homozygous recessive and it's a recessive allele here, then that means that, and this person is affected, it had to have gotten one from each parent, which means the other allele is dominant. Therefore, this one applies. So it's the recessive allele with both parents heterozygous. Okay, then we have done enough pedigree diagrams, and okay, we can actually, let's do one more. Okay. Now, this one is telling us a dominant allele causes the large joint of a little finger to bend inwards towards the 4th finger, capital letter B, and is called the bent little finger. The recessive allele, small letter b, causes the little finger to be straight. The pedigree diagram below shows the inheritance of a bent little finger in a family. Okay, so in this one, we have a pedigree diagram once again. and it is not sex-linked. How do we know it is not sex-linked? Because they indicated to us the alleles over there, and you can see that it is not sex-linked. Sometimes they also use the word autosomal dominant allele or autosomal. Then you know that it's also not a sex-linked inheritance. You need to know about color blindness and hemophilia. You need to know that those two are sex-linked. But if there's any other sex link that they're going to ask you in exam, they're going to tell you that it is sex linked, or they're going to give you the allele, which is going to indicate to you that it is sex linked. So this one is not sex linked. So let's have a look. Let's count the generations just because, right? This is generation 1, generation 2, generation 3. There are 3 generations. Okay? Then we've got the shaded ones are the males with the bent finger, and the unshaded ones are the males with the straight finger. Explain why individuals A and B are definitely heterozygous for this trait. Okay, so we have number A and B. They told us over here that the recessive allele is a small b, and it causes the finger to be, the recessive allele causes the finger to be straight. So this one, let's just look at it, the straight fingers. So these ones are all going to be small letter b. I like to put them in so that we already know that the recessive allele is the one that's going to be straight, because now we always think the affected, and we've got affected in our heads, and then all the shaded ones become homozygous recessive for us. But this one is not like that. Okay. So now they're saying that we have parents A and B. They are definitely heterozygous for this trait. How do we know this? Because the parents have the dominant phenotype. Okay, they've got the dominant allele. So they've got they've got the bent little finger. So it says that they've got the bent little finger. So they definitely have a capital B. And because they have children that have homozygous recessive, each allele must come from they must get one allele from each parent. Therefore, the parents have to be heterozygous, because then they can get this one and then they give this one. So you just say it like that. The parents have the dominant phenotype. To have children with the recessive characteristic, the other allele of each parent must be recessive. Doesn't have to be in this order, your bullet points, as long as it makes sense. Individual C has a child with a partner that has straight little fingers. Use a genetic diagram to show the possible genotypes and phenotypes of the child. Okay, so let's first see who is number C. So here we have part number C. It's female with a bent little finger. Okay, and then we've got, and they had a child with one with straight fingers. Okay, so we have bent little fingers is the phenotype, with straight little fingers, and the genotype is going to be heterozygous and homozygous recessive. Right, so we work out the gametes, we do fertilization, or you can do, you can either do the cross or you can do the Punnett square, and then when we get them, we get capital B, small b, homozygous recessive, and then we get heterozygous. So we have 2 heterozygous and 2 homozygous recessive. Okay, bent little finger and straight little fingers. Okay, so now we are going to go into a 2-minute break. Okay, just stick with us, and then we're going to be back in 2 minutes. Welcome back. Right, so let's just go quickly through what we covered today. We did dihybrid crosses, right? And we did pedigree diagrams. What I wanted to say before we went for a break is that these two topics, they are not easy, okay? They are difficult, they are challenging. However, if you practice enough and you go through enough examples, you will be able to be fine. We have a very jam-packed session today. We still need to go on to another important topic, that is also not easy. So we need to carry on, but there are lots of examples. You also have a worksheet that you can work through. You can get the PowerPoint presentation from your teacher. All the answers are there so that you can work through the actual questions. Okay, but practice, practice, practice. That's the only thing with genetics. Okay, so let's go through the next part, which is the mutations. Okay, we've got the mutations, and then we've also got genetic engineering that we need to do. So let's get into it. Okay, so remember we looked at our examination guideline. This is what you need to know on mutations. Right, so before we get into the exam tips, I want us to take note of these three definitions. Very, very important. Okay, you need to know what a mutation is. You need to be able to define a mutation, a gene mutation, and a chromosomal mutation. So the mutation is a sudden change in the genetic composition of an organism. That is a general description of a mutation. A gene mutation and chromosomal mutations, they are examples of mutations. Okay, so the gene mutation is when you have a change in the sequence of the nitrogenous bases or nucleotides in DNA. And a chromosomal mutation is when you have a change in the normal structure or number of chromosomes. Okay, so the way I remember it is that the gene has got to do with DNA. Okay, so therefore it's going to be a change in the nitrogenous bases or the change in the DNA. And the chromosomal mutation, chromosome, it's got to do with the number of chromosomes, whether it is more chromosomes that the individual is going to have or less chromosomes. Okay. Now the exam tips. Know the difference between the terms mutation, gene mutation, and chromosome mutation. Okay, you have to, because they're going to be specific when they ask you a question. They're going to ask you, define a mutation, define a gene mutation, or define a chromosomal mutation. Or they can give you a scenario about the mutation and they can ask you, what type of mutation is this? Then you must be able to answer that it's a gene mutation or a chromosomal mutation. The definition of a mutation is more general than the definitions of a gene or chromosome mutation, which are more specific. And also take note that the description of the point and frame shift mutations are not required. So you will see some older textbooks are mentioning these words. However, you do not need to know these and you don't need to know how they work. There are three types of mutations, okay, besides the gene overall, like a big term for our mutations. We have three big terms to describe them. We have harmful mutations, harmless mutations, and useful mutations. Okay, so whenever we think of a mutation, I think we automatically just go to the fact that mutations are bad. Okay, and when you look at some of the chromosomal mutations we're going to look at, and we look at the genetic mutations that we're going to look at, you will see that the ones that we are doing in our syllabus are harmful. Okay, that's the first one. Let's go through it and then we'll have a look. So I've divided it into cause, effect, and example, okay, to make it easier for you. So the harmful mutation is when there's a change in the DNA responsible for the production of a specific protein. Okay, a change in the DNA responsible for the production of a specific protein. So it's got to do with the protein making. And what does it cause? The effect of this. What is the effect of this? It causes changes to the organism's physical appearance or functioning due to an incorrect protein being made. Okay? And the example of these genetic disorders are hemophilia and color blindness. So as you know, these are harmful. Okay? So this is when there's a change in the DNA and it's causing a problem with the ones that code for the normal functioning of our blood clotting factors and the normal functioning of our rods and cones and our color vision. Okay? Then we've got the harmless mutations. These are the ones that occur in the non-coding DNA. So we have some non-coding DNA in our bodies, okay? And this DNA is not involved in making proteins, right? And it does not affect the structure or functioning of the organism. So this is changing the code, but it's not affecting our functioning or the structure. And this is, for example, freckles, blonde hair, or baldness. Okay, so they're actually a result of mutations. However, they are not harmless to us. Okay, they don't affect anything, but they are there. Then we've got useful mutations, and these are the change in the DNA responsible for the production of a specific protein. And protein made can increase the organism's chance of survival. And when the gene is passed on, it can lead to genetic variation. An example of this is natural selection. So this we are going to do in evolution. Okay, so you can just bank it for now. But I want to explain something about it to you, because we're going to do it a little bit later just now as well. So in this one, it's when there is a, this is when you are changing the DNA and it's going to change a protein. But what's happening is that when this protein changes, it can develop a characteristic that is going to be useful for the species. Okay, so it's going to be useful and it also can cause genetic variation, because what does genetic variation mean? Is the fact that we are all different. We did it in meiosis. Okay, we did genetic variation in meiosis. Remember, we had the crossing over taking place, which where genetic material is shared, and that's what basically ultimately causes us to all be different, and that all the gametes that are produced are genetically different. So these mutations are also good, because they can also, it could be a mutation that occurs that can be useful for the organism, and then it gets passed down over the generations, and then it can add for our genetic variation, so that more organisms ultimately are going to have that good characteristic. Okay, so we will do it a little bit later when we do natural selection evolution, so you can remember it for them. But they like to ask this question lately. They like to ask what type of mutation it is, and they want you to answer that it's a harmful mutation, harmless, or useful. I think the other word they use here is neutral, but the exam guideline is using useful and harmless and harmful, so let's make sure that we know them. Okay, right. Now, what are the examples of mutations? We've got the gene mutation, right? We spoke about it. It's a change in the sequence of the nitrogenous bases or nucleotides in DNA. And we have one example, which is hemophilia over here. We did it in detail earlier in the last session, so we're not going to go through it again. But you need to know that there are two examples of a gene mutation, the hemophilia and color blindness. They can give you any other one, but there will be an indication in the extract that they give you, or in the scenario that they give you. There will be an indication to you whether it's a gene mutation or a chromosomal mutation. Okay, they'll say something about the change in the sequence of the nitrogenous bases or something. Then example of a chromosomal mutation is one that you also all know that we did when we did meiosis. It is Down syndrome, and it's due to the extra copy of chromosome 21 as a result of non-disjunction during meiosis. Okay, so as we know with number 21, you can see there that there are 3 pairs and of chromosomes, and that is a chromosomal mutation. Okay, now we're going to do a quick activity. Now I want you to notice something about mutation activities. When they ask you questions on mutations, it's a bit sneaky sometimes. It's not just straightforward, like when you can see a question and you know this question is on a monohybrid cross, this question is on a dihybrid cross, this question is the pedigree. When it comes to this, they put it into other places. Because let me remember what I told you. I said that we have gene mutations and we have chromosomal mutations. So if you're going to have the gene mutation and you know that it's got to do with the sequence of DNA, you're probably going to see it in a protein synthesis question. So they'll ask you questions on protein synthesis, and then they will sneak in a gene mutation question. Or they could ask you something on meiosis, and they've got Down syndrome happening, and then they could bring in the chromosomal mutation there. Or they could give you an extract question on a new type of mutation that you don't know about, and they can give you information about it, and they can ask you application questions. So it's not like you can just go through past papers and say, I'm going to do all the questions on mutations now. Okay, similar with genetic engineering, but genetic engineering is a little bit better, a little bit easier to identify. Okay, so I just want to bring that to your attention so that when you practice, you need to be able to identify these questions. The diagram below shows the stage in protein synthesis. Okay, so it's protein synthesis, and just to remind you, let's go back to term 1. Which phase are we looking at here? Is it transcription or translation? And it is translation. Yes, how do we know? Because we can see tRNA, we can see mRNA, we see codons, we see amino acids, we see peptide bonds. Okay. So this question was extracted from another question. They asked a lot of things on protein synthesis, but we are only focusing on the mutation. So they're saying here that in codon 3, guanine was replaced by cytosine as a result of a mutation. So here we go, this T over here, it used to be guanine. Now it is cytosine. Explain how the mutation will affect the protein that will be formed. So when you get a question like this, you work it out first, okay, before you say how you're going to put it into words. When you put it into words, you're just going to basically put in what you did. So the first thing you have is that if it is UCA, okay, what protein is going to be formed now when it is UCA? Okay, so if we got codon, if the codon is UCA, then the TR, the anticodon will be, so we have UCA, the anticodon must be, I'm going to write it here because we're going to write other things, UCA. So the anticodon is going to be AGU. Okay, so if we go to AGU, it's supposed to be serine, right, or serine. That is what it's going to be now. But if it was UGA, which was the original, then what would the anticodon be? It would be ACU. And when you've got ACU, we now have threonine. So now the serine is going to be replacing the threonine. So the serine will now be replacing the threonine because threonine was there. And now it's going to be replaced by that. So then you say that. And then what is that going to do? It's now going to form a different protein. Why is it forming a different protein? Because we're forming a different amino acid. But the point I want to make for you here is that you need to say, you need to be specific. You cannot just say another amino acid is being used. Or you cannot say that now we've got, you cannot just say another protein is being formed because we have a different amino acid. You need to be specific to what the amino acids are. Same with if you're going to talk about the coding. Like here we said UGA codes for threonine and UCA codes for serine, therefore forming a different protein. Okay, so I just want to indicate to you that you need to be specific. Now this question is a different kind of a question. Here we have an extract, okay? Please, grade 12s, go through extract questions. You need to practice extract questions. In every paper, there's an extract. And there's reading for you to read, and learners struggle with this, okay? You need to read with understanding and know what you are reading, be able to underline keywords, read the questions, then read the extract again so that your mind can start working out what the questions are that you're going to need to answer. Right, so let's quickly go through this. Researchers have discovered that members of a particular family have high bone density that may be caused by a gene mutation. High bone density reduces the risk of bone fractures. 20 members of the family had their bone density measured and DNA samples taken. 7 had high bone density. The high bone density occurred throughout their bodies, but especially in the spine and the hips. Okay, this is a serious story. Right. Now, we need to describe what a gene mutation is. Now, you see, they're telling us it's a gene mutation. You need to describe what a gene mutation is. It's a change in the sequence of the nitrogenous bases or the nucleotides of DNA. Okay, so the key word here is that we are changing the sequence, and the other key word is that it's of the nitrogenous bases. Very important. You cannot say it's a change in the sequence of DNA. Because what about the DNA? Okay, it's about the nucleotides or the nitrogenous bases. That's where the actual change is occurring. Explain why it is necessary for the researchers to collect DNA samples. So why are they called collecting the DNA? It's because they have to see where the mutation is. Because remember, it's telling you it's caused by a mutation. So they need to check the individuals who have the high bone density. They need to check that the individuals that had the high bone density have the gene mutation. Okay? It's not just that they have the high bone density. It's the fact that they have the bone density and have the mutation. State the effect of this mutation. Okay, so now when they want the effect of something, they want you to say, what is this going to cause? Okay, so they have this high bone density problem. What does it cause? And it produces the mutation, what does the mutation cause? It causes the production of the high bone density. You can also say that it's useful or beneficial. Now, that's from the previous word that we were using. Because, yes, it's high bone density, but it's a good thing. It's a useful thing. It's a beneficial thing. So it's a useful mutation. Okay? And then the last question here we're going to do, you calculate the percentage of family members who had normal bone density. Show all your working. So they said we had 20 people. And we said that 7 had high bone density. So they want to know how many are normal. So we have 20 minus 7, which is 13. So we have 13 out of our 20. There's 1 mark for that. Times by 100 to get a percentage, and we get 65%. So 65% of the family members had normal bone density. Okay. Whenever you have calculations, please make sure that you have it. You show all your working, okay? This 20 minus 7, there's no mark for it, but you're not going to be penalized if you do work it out. So please work out whatever you need to work out. Just be able to have it make sense and in logical order, and you will get your marks. Okay, so like I said, mutations, sneaky. They can bring it in wherever. So you need to be able to answer questions like this, do enough past paper questions to be able to answer questions on mutations. Okay, then we move on to biotechnology. Biotechnology is an umbrella term for, let's first look at the definition of biotechnology, right? It's the use of biological processes, organisms, or systems to improve the quality of human life. Okay, so in this, there are basically, it's being done in the lab, or it's being done by scientists. Okay, and we are basically altering, they are changing the organisms, how they work in order to benefit or improve the quality of human life. Okay, so this is an umbrella term. Umbrella term means that it's like a bigger term, and then under this term we have other terms. So biotechnology is our broad term, and then under that we need to learn about genetic engineering. And genetic engineering is a process where scientists alter, swap, or manipulate the genes on the DNA to produce an organism with desirable characteristics. Okay, so in this one, they are now swapping and manipulating. This is the actual process of them swapping and manipulating the genes of the DNA so that they can produce an organism with characteristics that they want. For example, sweeter fruit, okay, or pest-resistant crops. Okay, those are characteristics that we would want, right? So they alter the genes in order to achieve that. Right, and this process is falling under a bigger process which is called biotechnology. Biotechnology is like the general term, and then genetic engineering is the form of biotechnology. Now, the 3 types of genetic engineering that you need to know are genetically modified organisms, GMOs, stem cell research, and cloning. Okay, now all 3 of these things are constantly changing. Okay, remember, science is not set in stone. All of these are constantly changing. So when you are reading, when they are giving you an example in the exam, it's not necessarily one that your teacher did with you in class. So you need to understand what GMOs are, what are stem cells, and what cloning is. Okay, so you need to know a brief description of these, and you need to know the advantages and disadvantages of genetic engineering, and you must be able to answer application questions on these. So what are the advantages and disadvantages of genetic engineering? Even though we haven't gone into the details yet, you can think for yourself, right? They are altering the genes in order to change the organism that they have the desired characteristic. Who is the desired characteristic for? It is for humans. Okay, so let's think now. What would be the advantages of this? Why would we want this? We can produce medicines, okay? We can control pests with specific genes inserted into a crop. We can use specific genes to increase crop yield or food security, and selecting genes to increase the shelf life of plant products, okay? People would want this. Why would you want this? You don't want your things to rot, okay? You don't want to run to the shops all the time. If it increases the shelf life, you don't have to go to the shops often. You can also make them have specific to have bigger crops, okay, bigger crops, better crops, so that we can increase food security. Now, they also have disadvantages. Remember, we are altering something. We are going against, some people believe that we are going against nature. So as they say here, it's interfering with nature, or they say it's immoral. The process of doing this is also very expensive, so not all countries can actually afford to do it. It has potential health impacts. We are unsure of the long-term effects, and new pathogens could be made for biological warfare. Okay, that's a scary one, because now they are developing things and they're making things and sort of man-made, and you can actually make disease-causing organisms and then use it for biological warfare. And lastly, it can also increase our loss of biodiversity, because now we are making the same crops, we are producing the same crops with the same characteristics over and over, or we are producing the same characteristics, so we can actually lose our biodiversity. Okay, so why I'm explaining these things a little bit more as well is that remember that they're not just going to ask you sometimes to list these. They're going to give you a specific scenario and you need to apply it. When they ask for the advantages and disadvantages, please make sure that you're as clear and precise as possible. Right, now we move on to genetically modified organisms. Right, so there's three. Remember I said genetically modified organisms, stem cells, and cloning. Right. Genetic engineering is used to alter the genome of a living cell for medical, industrial, or agricultural purposes. This results in a genetically modified organism, GMO, or transgenic animal with DNA from more than one species. GMOs are used to breed more productive crops or animals so that more food can be made, to produce drugs or hormones, example insulin, which have fewer side effects and is cheaper, to infect cells to cure diseases such as brain tumors and cystic fibrosis. Okay, so GMOs are mainly used for good things. So it's not only for food, it's also to make medicines, because that's how insulin is made. Okay, insulin, we've got insulin that is produced naturally in our bodies, and now we also have synthetic insulin that diabetes patients need to use when their pancreas is not working effectively. Okay, so these are some of the examples why we have GMOs. Now we move on to stem cells, right? What are the sources of stem cells? You get embryonic stem cells, you also get it from bone marrow. Okay, why do you use them? What do they use them for? They use it to treat cancers of the blood, example leukemia. They use it to replace dead cells in the heart after a heart attack, growing skin tissue to treat burn victims, growing nerve cells to treat spinal cord injuries and Parkinson's disease, and growing pancreatic cells to treat diabetes. Okay, very important. Basically, what I want to show you in this diagram, this is a very nice diagram, okay, but it's basically telling us here that we've got some stem cells here, okay, in this petri dish. And then what happens is that these stem cells can basically, remember the stem cells are it's like your normal undifferentiated cells. And then those can be differentiated. So that's what they basically do in the process, because now that's what they do in this process of the biotechnology involved, or the genetic engineering involved with this. So as you can see over here, from these, they can actually identify drug targets and test potential therapeutics. They can, the bone marrow they use for leukemia and chemotherapy. We have tissue cells, the tissue cells that they can get and harvest. They can use it for transplantation. The bone marrow they can use, nerve cells and for Parkinson's and Alzheimer's, heart muscle cells for heart disease, pancreatic islet cells for diabetes. Okay, so the other thing they can do is that they can understand prevention and treatment of birth defects. Okay, so there are so many different things, and there's other things that are unknown. There are so many unknowns still, because they are still busy with this research. Okay, so that's all you need to know about stem cells. Now, here's a little activity on stem cells, so you can see how you can actually apply this. When a child is born, the umbilical cord is cut and stem cells can be obtained from it. Many people think that the stem cells for treating human conditions should be obtained from umbilical cords rather than from human embryos. Recently, stem cells have also been obtained from bone marrow. These stem cells are used to treat conditions such as heart disease and spinal injuries. Name 3 sources of stem cells mentioned in the extract. Okay, so they speak about the umbilical cord, they speak about embryos, human embryos, and they talk about bone marrow. Explain why the characteristics of stem cells make them useful for treating some disorders. Like I said, they are undifferentiated. They have the potential to develop into any type of cell. and they can replace affected or defective cells. Name one condition in the extract that can be treated with stem cells. They talk about heart disease and they talk about spinal injuries. Can you see over here that question 1 and question 3 come straight from the extract? Question 2, it needs you to explain, so it requires a little bit more application. But there's definitely marks to be had here, and there are learners that are not getting the marks that come straight from the extract. So we really need to have a look at that. Now we look at cloning. You need to know a brief description of cloning, but let's first talk about, okay, let's look at it. The process that you need to know. Okay, so what you need to know over here is that we have a female sheep. Okay, there's a female sheep. There are 2 female sheep. So you can see there's a light one, a white color one, and there's like a light brown color, a beige color. And a body cell gets taken from this sheep. This sheep A is now giving its body cell. And they take the DNA out of that body cell. So basically they want to clone. In this diagram, we want to clone sheep A. So we take a body cell from sheep A. They take out the DNA from sheep A. And now the DNA is there. Right? It's extracted. Now we go to sheep B. What's happening in sheep B is that they take an egg cell from sheep B. And then they remove the nucleus. Then they take the DNA that was extracted from sheep A and they insert it into the cell from sheep B. Okay? And they fuse it. Sheep A fused with egg cell from sheep B, the DNA. They fuse it and now we've got the structure. Then the fused cell develops into an embryo, which is placed in the uterus of a foster mother. Okay? So then they've got a sheep C now. So the one sheep they use for the DNA because they need to clone that sheep. Why are we using the DNA from sheep A? Because we want to make another one of sheep A. So all the genetic material has to be the same. So we need to take a body cell. Why do we take a body cell? Because the body cell has all of the genetic information. Okay? All the genetic material. They extract it and they're putting it into an egg cell. Okay? Why are they putting it into an egg cell? Because it's a gamete, right? And then it's going to then fuse. Now they use these 2 individuals and now they're going to put it into a third sheep, which is now sheep C. The fused cell develops into the embryo and then the embryo is placed into the uterus of the foster mother, or they call it a surrogate, which is now sheep C. Then the lamb is a clone of sheep A. Can you see that? The lamb, then that embryo is going to develop and it's going to develop and it's going to become bigger and bigger and it's going to develop into a lamb, and that lamb is going to look exactly like the original lamb, which is lamb A. Okay. In biology, cloning is the process of producing similar populations of genetically identical individuals that occurs in nature when organisms such as bacteria, insects, or plants reproduce asexually. What are the benefits of cloning? Therapeutic cloning can replace damaged tissue, example skin, heart cells, and bone marrow, so helping to save human lives. Genetic diseases could be prevented. Superior animals may be bred to improve food supply and quality. Research in any form improves skills and could open other avenues due to spin-off technologies which could help mankind in the future. Okay, so you need to know the brief description of cloning, you need to know the benefits of cloning, and you need to know how you can answer questions on this. Now quickly, the last 2 things we're going to do, the paternity testing. So when it comes to the paternity testing, they're using the DNA plus the blood groups, okay, to estimate the probability that the man is the biological father of the child. They need to use the blood groups plus the DNA profile they can use in order to do this. So we did this a lot in term 1, but we're just going to do a quick example of this to show you how they can actually ask you this for this type of section that we are talking about and we are dealing with now. Okay, so a family wanted to identify the biological father of a boy. The mother of the boy is known. Blood groups of DNA profiles of the mother, the boy, and the 2 males were used to determine paternity. The data below shows the results of the 2 procedures. Okay, so they have the blood groups here and they've got the DNA profile. The question is asking, explain why paternity of the boy could not be established using blood groups only. Okay, so the reason why is because let's look at the blood groups. The mother is blood type O. Okay, the Mother is blood type O. The boy is blood type B. And there are 2 males. And if you look at this, remember that the boy has to receive 1 allele from the mother, 1 allele from the father. So we definitely know that this boy is heterozygous. Why do we know that? Because the mother is homozygous recessive. And 1 allele must come from the mother. So she had to have gotten the small I, the recessive allele, from the mother. Which means that that one must come from the father. But now here's the problem. Male 1 and male 2 both have the allele for blood type B. So either one could be the father. You see? That's why they need to now use the DNA profile to check the DNA bands, because we can't only use the blood type in this instance. So now you need to explain it just like that. 5 lovely marks. How do we start? So you first say either male 1 or male 2 could be the father of the boy. Why are we saying that? Because both males have the B allele, right? You can go further to say that male 1 is AB. You can write the genotype, and male 2 the possible genotypes. You can go further to write that. The mother's blood group is O and must have the genotype homozygous recessive. The boy would have inherited the recessive allele from the mother, and he would have genotype, which is now heterozygous. Okay, so you just need to be specific, say everything that you need to say, and then you will be fine. Who is the biological father of the boy according to the DNA profile? So now you look at the DNA profile. What are you going to do? You're going to match all the bands with the mother. All the bands with the mother, those match, these ones match, and this one match. The remaining bars must match with the father. So this is male 1, male 2, sorry. This one is also matching with male 2. This one is also matching. This one does not match with male 2, but it's fine because we still have enough here that the remaining bands are matching. So we have male 2, definitely male 2. You can see we don't have enough matching with male 1. Explain your answer. 4 or some bands of the boy's DNA profile match with those of the mother. We did that. It was 1. It was, oh, sorry, the remaining bands. What am I? Sorry, this is a mistake. It's the boys. The remaining bands of the boy is going to match with male 2. So we've got this one. And then we've got this one. It's also matching. And then we've got this one that's matching. And we've got this one that's matching. And that one matches the mother. So we look at the boy. And we have to see how many of them match with the mother. And the remaining bands of the boy is going to match with the father. So now you need to be specific and count them. Okay, how many are here? 1, 2, 3, 4, 5, 6, 7, 8. 4 of the boys' bands of the boy match those of the mother, or the mother's profile. The remaining bands of the boy match with the band of male 2. Okay, so that's how you're going to explain it, right? And that's how you work it out. So can you see this is a question that we can actually did with, it could be a question that can be asked as DNA profiling, but it's also asked in this question in genetics because we are also bringing in the blood groups. Okay, very nice question. So you see how the examiners can bring in more than one thing. So you must be able to just keep your wits about you. Right, then the last thing we're going to do are the genetic links. This has got to do with mitochondrial DNA. So mitochondrial DNA is found in mitochondria. Mutations in mitochondrial DNA are analyzed by scientists to work out a timeline of genetic ancestry. Only the mother's mitochondrial DNA is passed to her offspring. They use the maternal line to compare mutations of different people and see how closely related they are. The more mutations are found, the older that race is believed to be. Okay, so let's quickly talk about it. Why do we have, so here's the cell, okay, and here's the mitochondria, and within the mitochondria we have mitochondrial DNA. But why are we saying that only the mother's mitochondrial DNA is passed to the offspring? The reason why is because during fertilization, only the head of the sperm penetrates the ovum. And where is the mitochondria in the sperm cell? It is in the midsection, in the neck. That neck does not enter the ovum, so it falls away. So the only thing that is left in that zygote that's going to form is going to be the mitochondrial DNA, because the ovum is going to have the mitochondria. Okay, So that's what it's saying over here. Now they're saying that, so they use the maternal line because they've got the mitochondrial DNA. They use the maternal line to compare the mutations of different people to see how closely related they are. So if we have to look at this diagram we have over here, nuclear DNA is inherited from all ancestors. You can see over here that the nuclear DNA is being passed down all the way, okay, from everybody. But when it comes to mitochondrial DNA, it's inherited from a single lineage. So you see that the pink area that's shaded out here, the pink shading is showing us that it's from this female, it's going to go down into the offspring. Then from that female, it's going to go down into the next generation. So that's how it's passed down through the maternal lineage, is the mitochondrial DNA passed down. Right, that's all from us for this week. It was a lot, and like I said, lots of challenging topics, and but lots of exciting stuff and very interesting. Please make sure that you do some more work on this, and you ask your teachers lots of questions and practice lots of examples so that you can be ready for our June exam, which is coming up around the corner. Good luck.